Good evening everyone. Today we are going to discuss about tooth development. Basically, tooth development is a complex process and has different stage. So we will understand it in simpler language. So first, this is oral epithelium and this is dental lamina. The oral epithelium and the dental lamina is separated with a very thin basement membrane. So we have different stages of tooth development. First is bud stage, then cap stage, then bell stage, and then advanced bell stage. So we will understand it one by one. So first is bud stage. The oral epithelium, here it is ectomesenchyme. So ectomesenchyme stimulate the oral epithelium to form the dental tooth. So this ectomesenchyme stimulate oral epithelium to form a bud-like shape in the oral epithelium. These are 20 in number in case of children. So these bud-like shape in future forms the 20 tooth, that is milk tooth. So it forms the bud-like structure in oral epithelium. So the first stage is bud stage. Here there are two type of epithelium, low columnar and polygonal cell. Low columnar cell are present in the periphery and in center polygonal cells are present. Polygonal cells and outer low columnar cells. So in bud stage we have two epithelium, two type of cells, polygonal cells, that is the central cells, and the low columnar cell. So second stage is cap stage. In cap stage this bud form a cap like structure. Here from the oral epithelium it form a cap like structure. Here the outer cells are low columnar cells and inner cells are differentiate into stellate reticulum. So in cap stage we have two type of cell, low columnar cell, that is outer peripheral cell, and the inner stellate reticulum, which is star shaped shell. Then coming to third stage, that is bell stage. In bell stage this oral ectomesenchyme, which is now known as dental papillae, which further form dentine and pulp in future, it stimulate the low columnar cell to form the ameloblast and further enamel formation. So in bell stage the oral epithelium, which was initially cap like, will form the bell like shape. Here three types of cells are present: outer low columnar cell, inside stellate reticulum cells, and now stratum intermedium. These third cells are also present. Fourth stage is known as advanced bell stage. In advanced bell stage the tooth is completely formed and here the enamel deposition occur and the dental papilla has converted into dentine and pulp and the tooth is completely formed. So this was the different stages of tooth development. So in simultaneously with tooth development, there is some different process going on, or we can say the different stages of growth of tooth development is going on histopathologically. So histopathologically we have different stage like initiation, proliferation, histodifferentiation, morphodifferentiation, and last is apposition. So these are the different histophysiological stages. We have already discussed about the morphological stages of the tooth development. This is histophysiological stages. So the initiation, the first stages is coinciding with the first stage of the tooth development, that is formation of dental lamina. So as I have already told, first the oral epithelium, then dental lamina is formed. In first stage, bud-like shape is formed. So the first stage is initiation, in which dental lamina is formed. So any disturbance during this stage will lead to the disturbances in the number, because as the name suggests, initiate, so it causes disturbance during the initiation stage of the tooth development, like number of tooth. The tooth may form less or the tooth may form more. Then the second stage is proliferation. Proliferation stage occur in bud and cap stage. So when the tooth development goes in bud and cap stage, and here the disturbance will lead to the disturbance in size and shape. As the name suggests, proliferation, so meaning during proliferation there is, if there is any disturbance, then it will lead to the difference in size and shape. Like the example is, if there is disturbance in the proliferation, the tooth may form twinning, fusion, two tooth may fuse during the development stage, and incomplete division of tooth into two tooth, like that. Then third stage is histodifferentiation. In histodifferentiation, it occurring during early Bell stage. So early Bell stage, we have three type of cells. So disturbance in the histodifferentiation will lead to the difference in structure, like histologically, amelogenesis imperfecta, dentinogenesis imperfecta, like that. Next is morphodifferentiation. Morphodifferentiation, it occurs during the early and the late Bell stage. Early and late. As the name suggests, morphodifferentiation. If there is disturbance in the morphology of the tooth. The shape of the tooth is changed, like paglateral, mulberry molar, congenital syphilis, like that. The last stage is apposition stage, in which there is disturbance during the formation of enamel and dentine matrix, and disturbance during this stage will affect the structure of the tooth, like hypocalcification of the tooth, enamel fluorosis, etc. So these are the different histophysiological stages.